Welcome to Lecture 4 of Evolutionary Genetics, ZOO3649 2020. And today we're going to be talking about bioinformatics, more about bioinformatics, and today more about data generation and databases. What do we do with all this data we're generating? By data, what am I talking about? It could be DNA sequencing data, whole genomes. One whole genome is a massive sequence of DNA, and so this is a huge amount of data. It has to be stored on a disk somewhere, not just generated. So there are other kinds of data which we'll get into later in the lecture. But I just put this slide here up to show you that next generation sequencing, these machines, each one is a high-seq X, and each one is capable of sequencing 320 human whole genomes per week. So you can imagine this has already been this state of affairs for the last 4 to 5 years. So you can imagine how many genomes have been sequenced since then. If it can do 320, one of them can do 320 a week. So at BGI, Beijing Genomics Institute in China, they have a massive warehouse with hundreds of machines like this, each one of them capable of doing 320 human genomes a week. So you can imagine how much DNA sequence data is being generated for how many different animals, plants, viruses, bacteria. So this graph here, you see, since 1980 to 2000, how many nucleotides have been sequenced. And you see that since the 90s, it has undergone an exponential increase. And this is only 6 billion nucleotides, okay? That's about the amount of 2 genomes worth. So you can imagine with 100 genomes, how much higher this red line is. It's probably, if you extend the red line, it's probably going all the way to the sky right now on this scale. But DNA data is not the only data that we're generating with NGS. So with NGS, we can also generate RNA data. So with microarrays, we can quantify RNA levels of gene expression in the genome. We can look at different genomes in parallel, transcriptomes of different genomes in parallel, and we can basically add this RNA data to our existing databases, so all these transcriptomes. The other kinds of data are proteomes. So when we look at a protein and we want to look at the building blocks of the protein, so that's called mass spectrophotometry. And so these mass spectra libraries are also giving us a lot of data about proteins. So it's not just DNA sequences that are responsible, and NGS that is responsible for this huge trend in data growth in the last 10, 20 years. There's also metabolomics. Okay, so all of these things, as you see, end with omics. Okay, these are the omics biotechnology new buzzwords. So metabolomics is looking at metabolites, and as you know what metabolites are, and they look at how these metabolites are characterized and how secondary metabolites are characterized, and you end up with these mass spectra comparing different metabolites, and all that data also needs to be stored somewhere. We've just been generating so much data. In fact, this little drawing here is kind of putting it into context. It's a bunch of old school scientists sitting around a 3 billion piece jigsaw puzzle. So this is like a jigsaw puzzle. Normally there are like 1,000 pieces or something like that. This is a 3 billion pieces jigsaw puzzle, and you know why it's 3 billion pieces? Because there are 3 billion nucleotides in the human genome. So basically, this is the human genome in the style of a jigsaw puzzle. Now you can imagine you need a very serious computer to take each piece and figure out and put them all in one line and figure out which piece fits in which sequence. And that is why we have bioinformatics, to deal with this enormous amount of data that we've been generating. Okay, bioinformatics, and not just bioinformatics, you also need the internet. So let's look at the bioinformatics part. Bioinformatics is not something that's new. It's actually been around for a long time. But what is it? It is basically using computers and algorithms, which are basically little programs written in the computer. And these algorithms that work on these computers help us scientists to access, to process, to store, to share, to retrieve, to visualize, to annotate our data. Okay? So we use these algorithms for that. And what we also need are very, very fast computers. We use servers at the University of Venda. In fact, everywhere that does genomics analysis uses big computer servers. This here, I put this picture in as an example of an old server from the 1960s. This was the Margaret Oakley Dayhoff protein database, and it was the first program or algorithm that was used to actually to assemble DNA or line up protein sequences. And you see here these big computers here, they're using as their storage actually a tape, a round tape with a ribbon tape on it, and that was what would record the data. And so I'm very glad to say, and each one of those tapes could probably store much less than what your mobile phone today can store. So you can imagine how much more computing power we have these days with our modern servers compared to back in the 1960s. So our computers have gotten much better, but what's also then gotten a lot better is the ability to store the data. And they are stored in databases, right? So now when you are doing research, when you're looking for biological information, you don't have to start from scratch. You don't have to figure out a lot of the information for yourself, because why? A lot of information is already there. It's already stored in these biological databases. And we, unfortunately, because of COVID, we are unable to do the lab, the practical, that shows you how to access these databases because of our social distancing. We're only allowed a few labs. We're only allowed to do a few labs per module. Let me just turn my sound off. There you go. Okay, so what kind of databases do we have? We have databases for pretty much everything. And we have many, many different kinds of databases. I've listed here the most popular databases that are used by us in our field of research. But to be honest, there are hundreds of databases out there with a lot of information. One example, one of the biggest, is the NCBI, which is the National Center for Biotechnology Information. That is a United States database, and it's a United States government database. But everybody, all nations, scientists from all over the world store their data in that database. Then there's the Dryad database to store project-specific data. There's UniProt or SwissProt for protein sequences. So, and that's just to name a few. Yeah. And what can you? So, if you want to find out something, say about a particular gene in the genome, what can you find out? Depending on the gene and how much information you have available, you can find out evolutionary information. You can find out genomic information, so where in the genome the particular gene is, what shared domains, what regulatory regions are involved with that gene, what structural information, what kind of proteins it produces, how expressed, what kind of expression profiles you have, and expression information you will get from, of course, RNA-seq data, okay, transcriptome data. Structural information you will get from protein data. Genomic information you'll get from NGS data. Evolutionary information you'll get from genomic and all of the other datas analyzed together. And you can also find functional information. So what enzyme, what pathway does that gene form? What role does that gene have in the cell? in disease, etc. So depending on the gene that you're interested in, you can find out a lot of information that's already there. Information that you don't need to find out for yourself. It's someone else already found it out, and all you need to do is search the database to find out for yourself. So how do we get information out of a database? Well, we can browse, okay, the way you would browse the internet. There's no target, but a much better way is obviously to search for particular information. So when you're searching, what do you put into the search? It's like searching for anything. If you read a paper, for example, and you don't understand the paper, well, you're going to have to search for keywords in Google about that paper. So in a database, say NCBI or UniProt, you are looking for a particular protein. Of course, in the search, you will say not something like protein, but you will try to give the name of the protein. Okay, so you need to have some kind of way to identify, like the name of the gene, the sequence of the gene, or other information. Okay, and it helps to have particular goals. It helps to, when you're searching a database, to know what it is you're actually looking for. Okay, you can also find that genes and gene product database are organized by their sequence. So the actual genomic sequence that encodes the gene itself. The gene products are because sequences for genes are unique, the gene products then can be uniquely identified by the sequence. So you can actually search for a gene, not by naming the gene itself, but by actually having the sequence or part of the sequence of the gene. You can paste that into a search, go enter, and it comes up with all the information about that gene. So that's fantastic. You don't have to know the name of the gene. You just have to know the sequence. If you don't know the sequence, you need to know the name. Okay? So macromolecular sequence provide biological meaningful keys for searching databases. Okay? So keeping on the searching of databases, you need basically to use a sort of key, right? And I just explained to you now what these keys would be. You could have the gene name, the sequence name, and many kinds of inputs, whether amino acid, nucleotide, messenger RNA, cDNA, complementary sequences. You can put in any kind of sequence. Okay? So once you hit enter, what happens in the search algorithm? You're looking for something. What happens is that an algorithm is called. So your search uses an algorithm, which is a little app, a little program, and it uses algorithm to search through the database to retrieve information that is similar to what you've given it. Okay? So exact matches are rare, but it doesn't have to find exact match. It can find a similar match, which is often what you're actually after. Okay? We can want to know a lot of things about a DNA sequence or about a gene. Okay, we might want to know how similar the sequence of the gene is to other known genes, which is its closest relative. What is the significance of the gene, of that sequence? Okay, does it have a functional role? So we need to know genomic information. We need to know structural, functional information, and evolutionary information. So this is, again, as you realize, this is repetition. This lecture from 2544 last year, and it's also repetition from a couple of slides ago. Okay? And this repetition is to make sure that you understand why these databases are important and what kind of information you can get out of these databases. Okay? So that's the bioinformatics part. What about the internet part? Remember, the answer is the bioinformatics, which all the algorithms and the big computers, but also the internet. Why? Because these omics projects, you often will need to find huge amounts of data which are not stored on your computer at Univen. Okay? Your computer is only so big, you can't store all the data you need for maybe a bioinformatics project. And so how would you do that? The only way to do that is to then use servers, or computers that are based all over the world that have the information. And that way, when you go to NCBI and you type in your search, what is it searching? It's not searching your local computer. It is searching many other hundreds of computers in other parts, sitting in other parts of the world. Those computers have the information. Imagine when you go to YouTube and you search in for, I don't know, whatever you want to watch on YouTube. What happens? Where is that video? It's not on your computer. It's on the internet. It is on someone else's computer somewhere, and that computer is linked. So now you are able to enjoy YouTube when you yourself don't have those videos downloaded on your computer. Why? Because the power of internet. You are able to use those videos, see those videos, even if they are on someone else's computer, somewhere else in the world. Because of what? The internet. We're all connected to each other. Okay, that's how it works also for bioinformatics. All right, so we are connected with a lot of computers all over the world, and those computers have information that we want. Because why? Our computers cannot have all the information we want. Okay, they're not big enough. So that's why many computers all with little pieces of information. The amount of information, total information stored is huge, and all of that can be accessed by anyone inside who is connected to this grid here. Here in blue, you see the NCBI server in the United States, the EBI, European Bioinformatics Institute in Europe, one in Australia. And so they are connected to the internet. They are connected to my computer at home. They are connected to our local networks in our bioinformatics lab at Univen. They are connected to other labs at Univen. So you see, any one of these computers can access the information from all the others. And that's the power of the internet. Okay? So you are here, you are asking, you put a request in, you saying, I want to search this database. Okay? It actually then asks other computers to do various parts of the search, and all those other computers put it together and give you a result. Okay, that's the power of the internet. Okay, what about the value of bioinformatics? I think soon we will not be able to place a value on bioinformatics. Okay, bioinformatics was, current bioinformatics, this was already 3-4 years ago, 300 million per year. Okay, half of which is in software development. That just goes to show how important the algorithms are. Because without those algorithms, the bioinformatics, you can't get everything. You can't get anything out of all the data you're generating. You don't know how to store it. You don't know how to access the information. Okay, so the prediction in the next 5 to 6 years would be that the bioinformatics is worth more than $2 billion, United States dollars. I think that within 10 years, it will be worth 10 times more than that. Okay, already we are seeing 50 or more bioinformatics companies, all generating next generation sequencing, proteomics, mass spec machines, which are going to make data gathering even more incredible as we go further into the future. Okay, so that was your lecture on concluding the bioinformatics part, just to give you an idea of what is out there. We're not really going to be doing a prac on this, unfortunately, this year because of COVID restrictions. In the second semester, we are also coming into a bit of a second spike maybe now, so we have to be extra vigilant. So we won't be able to do a prac on searching databases. You're lucky. You're lucky. Okay, so meet me next lecture. Okay, in lecture 5, we will also be going through some information that we had in 2544, but we're going to go into more detail. So please don't miss the lecture. It is very important for us to be able to measure variation. Okay, and we're going to go through the ways in which to do that in the next lecture. Okay, so join me for the next lecture coming up in a moment.